Hi, my name is Shui, and I'm going to show you how to make millions of copies of DNA using a machine that's called the Thermocycler. There are two different versions of these machines. There is the standard benchtop machine, and there is a newer, more portable version that is called the mini PCR machine. All thermocyclers do the same thing. They automate the repeated heating and cooling cycle needed in order to make a new copy of the original DNA. In the mini PCR machine, you can see all the important components of a thermocycler. Now let's take a look at the inside of it. Here is the metal block, which you will place your PCR tubes, which is attached to a heater that will heat your sample up to a denaturation temperature of 94 degrees Celsius. Now below that are some cooling components. In this case, some very efficient fans. These fans will cool the samples back down to an annealing temperature of 55 degrees and 65 degrees Celsius. The samples then are heated again back up to a temperature of 72 degrees Celsius for the extension step. This completes one PCR cycle. The machine then will repeat this process 25 to 40 times. This PCR cycle is controlled by the circuit board, which is connected to the temperature sensors, the heater, and the cooling blocks. The heated lid will prevent evaporation from happening from the samples and also will prevent condensation from forming from inside the tubes. The PCR tubes, now with all the reagents inside, are ready to go into the machine. To open the mini PCR machine, gently press the tabs with your index finger, your thumb, while you lift the lid. Place the tubes inside the wells. Close the lid and use the top knob to adjust the heated lid so that it presses gently on the tube caps. This prevents the tube from opening during the reaction. To program the mini PCR machine, connect the USB cable to a Mac or Windows computer or a tablet, and then open the mini PCR app. Congratulations! You now have millions of copies of a specific DNA molecule inside each tube.